Hi, everyone. Welcome back. Now that you've had a chance to listen to Jean tell you about sequences of concern and Todd tell you more about SeqScreen and details about the different workflows and how it operates, I wanted to revisit this example SeqScreen output that we had looked at previously to tell you more about the different columns of output now that you have more context for what is going on and what's happening here. So to refresh your memory, this is an output from looking at a FASTA file of single gene sequences. So each row is a different query sequence. And previously, we talked about this BSAT hit column, right? So that was a binary yes or no according to whether or not the query sequence had hit to one of the sequences in our custom database that included all the publicly available sequence data we compiled according to taxonomic lists from the US or from Australia or the US Commerce Control List that describes organisms that should be regulated. So we talked about how the BSAT hit column is sensitive but not specific, and we looked at the multi-tax IDs confidence field to understand how sometimes there are ties where a gene sequence can be equally likely to have been derived from a select agent or one of its closely related microbes that's not pathogenic. And in that case, we're not worried about that particular sequence because it's not involved specifically in pathogenesis as it relates to that select agent. So we talked through that. And just moving forward on that thread, I think I'll mention that this tax ID field is another thing related to the multi-tax ID confidence in that some of our end users previously wanted to see a single tax ID. So they didn't want this long list. They just wanted one number that they could go from that was the most likely organism that sequence came from. So the most likely taxon of origin. What SeqScreen will do in this case is it's going to report the highest probability tax ID in this field. If there is a tie, it will just take the first tax ID on the list. So in this case, we see 1428 is 1.0. That's the highest probability that there can be that something came from that particular taxon. But oh no, there's other 1.0s, right? There's a lot of 1.0s. Since we only put one tax ID in this field, SeqScreen just randomly takes the first one on the list and puts it there. That's something to be aware of when you're interpreting these outputs, that if you only look at this tax ID field, you will be missing out on any ties that might have happened in the probability and the confidence here in column D. So do be aware of that when you interpret things. GO stands for gene ontology. So this is going to give you the gene ontology numbers, which correspond to specific labels. Those are not only for pathogenicity. These can be for anything. SeqScreen will by default report molecular function or biological process Go terms. There is an optional flag that I ran in the example command for this exercise called include CC that includes the cellular component Go terms. So in that case, cellular component Go terms will be reported here as well. But these have to do with all kinds of different biological processes, molecular functions, cellular components, so where in the cell these are going to take place. All kinds of things show up in Go terms, right? So this is a very broad collection of function. And we've actually used this in other studies to look at things beyond pathogenicity, just to collect functional labels for sequences and do other types of analysis. So that's an interesting angle here, but not something specific to biosecurity. Okay, this is a field that you can ignore. It's always going to say 1. Don't worry about that. That is nothing. And then here we have all the different fun socks. So for the purposes of this illustration, I added a filter to my spreadsheet, and I recognize that that does cut off these labels for the fun socks, which could be frustrating, but fear not, because when you open your spreadsheet, you will be able to see these labels. And if you want more information about what the fun socks are, I would definitely encourage you to look at the software documentation We have a wiki page dedicated to functions of sequences of concern. There's this handy table here that'll tell you the FUNSOC definition and more information about it. So please check that out if you want to know more information about the specific FUNSOCs listed in SeqScreen. And please also refer to the publications that Gene Godbold authored about sequences of concern and categorizing them, where he goes into more detail about all of these concepts. Related to that, there's also the PathGo terms, which tend to be more detailed and numerous than the FUNSOC terms, but the big differentiator between PathGo terms here in this column and FUNSOCs here is that FUNSOCs in SeqScreen were all derived from machine learning. So these are all assigned by machine learning, whereas these PathGo terms are assigned by subject matter experts, so humans and machines. We included both because humans and machines are both important for these kinds of applications. Humans can only annotate so many things. It's a time-consuming process. It takes some level of expertise. You have to read through the literature, find experimental evidence to make the different assignments, and that's after the ontology has been developed. The ontology itself can take some time to refine and review and create. So there's a lot that goes into the manual annotation and assignment. But once you have enough of those manual assignments, you can use machine learning to scale them and iterate and go through and improve it. And that's essentially the model that we've used here. We did manual assignments of FUNSOCs in our original SeqScreen publication. Those were then scaled with machine learning to assign the labels to many other things. So when we look here at these FUNSOC labels, there are zeros and ones, so binary, yes, no. A dash means that it was not determined, so the machine learning algorithm did not assign anything to it, 0 or 1. So if we look at ones here, and in SeqScreen, you won't see them in color. Again, I just added some color to my spreadsheet for the purposes of this course. Here's our ones, and we see that disable organ, which is the most severe FUNSOC. And here's a little expert tip on SeqScreen output. The FUNSOCs are roughly listed in descending order according to how severe they are, or the like the negative effect they might have in their pathogenicity potential. So disable organ is listed first because if something can shut down an organ, that's very bad. And so what we see here for disable organ are a lot of those eukaryotic toxins, right? That they can be very damaging, and the machine learning is flagging those as highly concerning. If we go to see if there are any PathGo terms associated with those, we also see, some different PathGo labels, and they're not going to be completely equivalent to the FUNSOC labels. For one, because they're just called different things in the PathGo ontology and the FUNSOC ontology, but mainly because the humans are going through and very accurately finding publications and references and making assignments here, whereas machine learning is more broadly just looking for evidence of how to assign these different terms. Okay, so that's disable organ. We see the eukaryotic toxins having that. We see that the BSAT hit column is no, because these are not on any regulatory lists, but they have functional evidence of being damaging. And then we see some PathGo terms here that talk about how those things may be damaging as well. And again, this dash here in the PathGo field does not mean that it's not a pathogenic sequence. It just means that the manual curation team did not curate that particular protein. That's all that means. So dash means we didn't look at it. It's also worth noting that the UniProt E values here are very low. These are strong E values. These look good. And when we look at the different UniProt gene names, these all look very reasonable based on what we know these query sequences were. So I think this is a solid result here for these particular eukaryotic toxins. This is pretty expected. All right. And then if we take a high level view, just seeing what else is here in FunSoc land, you can see my green ones indicate that we have a smattering of different FunSocs assigned to these different genes. And if we look at the overlap between BSAT hit and FunSocs, we see things like here's a dangerous gene from Bacillus anthracis, the lethal factor gene, and it's got some FunSoc assignments. It's labeled as lethal factor. I mean, the name just sounds bad, right? And then it also has patho terms that describe how we know it's functionally damaging to the host. So this is clearly something of concern. It's overlapping with the BSAT hit and functional labels that are concerning. So that's something that I would definitely pay attention to. And there are a number of other things in here. If you look at what I collected for this list, I collected a lot of things that overlap with the BSAT list, but a number of the genes aren't necessarily damaging. And the reason I did that was I wanted everyone to see how, often there's no FunSocs, right? This is common because most genes are not pathogenic. If you were to just take a random sampling of genes out there and run them through SeqScreen, you would see a lot of zeros, right? It's not common to see pathogenic qualities in a sequence. And in this case, we're looking at things from Clostridium botulinum. So obviously there's a big toxin that can be made there, but there's also just a lot of normal everyday housekeeping genes that aren't concerned. So that's something to kind of look through as you're learning how to interpret everything. It's also interesting to see how some of the different codon optimization sequences, codon optimization processes for different organisms. So basically adapting the codons to be optimized to express in different organisms didn't necessarily change results, right? These all look pretty similar, even though we've optimized them for different organisms, which I think is a good thing. It's showing the strength of SeqScreen reporting things consistently. Here we see different parts of a gene. So I basically broke this up into smaller pieces. There is an output here that tells you the size of the query. And so this is a fairly small query size, but we're still getting pretty good results with small sequence sizes. I would say that SeqScreen performs pretty well with sequences down to 50 base pairs. Lower than that, it can still return results. It's just the smaller you get, since we're looking at things and we're talking about coding sequences, you start to get into very short amino acid segments that can be pretty ambiguous. So 50 base pairs or above, SeqScreen should give you reasonable results. But feel free to look at as low as you want. Obviously, the longer a sequence is, the stronger the hits are going to be. And I think that's the majority of what I had to say, other than the machine learning process in SeqScreen is not run in real time. It's run ahead of time, and then we pre-populate our database with all of those results so that every time you run a query sequence through SeqScreen, you should get the same result. You shouldn't see different results each time. And the machine learning is not perfect. No doubt you will find instances where you think you disagree with the machine learning results for one reason or another, and that's fair. We definitely don't claim that the machine learning is a perfect system, but it is the best system that we could develop given the limitations with scaling manual curations. And we do hope that as we improve labeling and ontologies and we get more and more curations in databases that are high quality, our machine learning performance is going to be improved. So we have really high hopes for that in the future. And we're pretty happy with how this is performing now, but we definitely recognize it's not perfect. If I had a choice between going with machine learning results or these manual PathGo results, I would definitely take a close look at the PathGo results, because I know that those were assigned and reviewed by experts in the field. And then one other final thing I'll mention as we wrap up this is that there is an HTML report that I haven't showed you yet. So this is what the HTML report looks like. You do need to load the file before you view it. So when you open up the output file, you will not see anything. You'll just see a shell of an HTML framework. So you need to choose the file and then choose the particular data file that you want to load. All right. We will pause there and come back to this in just a moment. Thank you.